Good day, this is the Professor, CBMD PhD, and today's Friday, May 19th, 2023. It is 6:35 and 29 seconds, a.m. Eastern Daylight Time, and today's research, cancer cells. So let's hop to it here. As cancer cells use a new fuel enhancement of sugar, a new study shows that in mice, the metabolic uridine can feed pancreatic cancer cells when glucose availability is low. Date May 18th, 2023. Source: University of Michigan. Researchers have discovered a new nutrient source that pancreatic cancer cells use to grow. The molecule uridine offers insight into both biochemical processes and possible therapeutic pathways. The findings show that cancer cells can adapt when they don't have access to glucose. As pancreatic tumors have few functioning blood vessels and can't easily access nutrients that come from the bloodstream, like glucose, researchers explain that without the right nutrients, the cancer cells get hungry. They know they still grow, obviously, but what are they using to grow? These findings show that, under certain circumstances, uridine is one of those fuels. The scientific investigators say the ability of cancer to switch to alternative nutrients has fascinated researchers for a long time. Blocking such compensatory switches can lead us to new treatments, and that is the door we hope this study will open. Uridine is present in a tumor microenvironment, but its exact source and how cancer cells access it remains a mystery. Part of the picture is it is in the bloodstream, but we don't know where it is coming from. Specifically, likely it is coming from multiple places, and so far we haven't been able to pin it to a single source. Events that researchers refer to as times of crisis, when cells don't have enough nutrients because of limited blood access and/or intense competition between cells, could be a clue as to why and where cells turn to uridine. The scientific investigators say cancer cells seem to be sensing the concentrations of glucose and uridine in the local environment to inform their adaptation. They recognize the unknown regulatory process as well as a cancer-promoting mutation in the KRAS gene, which is common in pancreatic cancer, as two ways that cancer cells control the usage of uridine. They use the technology that screens hundreds of different nutrients to which ones support pancreatic cancer growth. Typically, researchers look at standard nutrients like sugar, protein, and fat. Researchers took an unbiased approach. They used a large panel with over 20 pancreatic cell lines and around 200 different nutrients to assess different ways pancreatic cells grow. What do they actually metabolize? And this method led researchers to discover uridine. And this method offers therapeutic insight too. The findings show that uridine is metabolized by the enzyme uridine phosphorylase 1. Blocking uridine phosphorylase 1 had a major impact on the growth of pancreatic tumors in mice. Findings that suggest the importance of testing drugs that block uridine as possible new treatment options. As there is potential to better understand and treat pancreatic cancer with new drug targets and new therapeutic approaches, more research is needed to determine the best way to move this discovery to the clinic. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.